Okay, so I think there's a few people who are in the LinkedIn Live. This is the first time that we're running something like this, so please bear with us while we manage the tech and all of the sharing and all of that lovely stuff. So yes, welcome to the session. The idea is that today we'll talk you through some of the new features that were in the release that was yesterday, which are hugely exciting. And here you have a selection from across the team who hopefully can help answer any questions that you might have. So once I'm done with the walkthrough, we'll switch over and we should be able to see the questions that you put in the comments and then answer those. As I said, please do bear with us as we manage this. Cool. So just how this session will go. So I think I've said this already, but we'll walk through the new data and the new features this release. If you have a question, please type it into the comments. If we see it while we're presenting, then we'll try and answer it. But if not, we will answer all the questions after the walkthrough. So I thought I'd start with a kind of very quick overview of the different features of the release. So to start off with, we had our last release, we merged OpenTarget genetics data and analyses into the OpenTarget platform, which means we can pull in for every release new data, new credible sets, which is hugely exciting. So in this one, we actually have 36% more credible sets than the last release, and that's from 211 publications, which includes over 170,000 credible sets from the Million Veteran Program, which is hugely exciting. The second kind of major thing in this release is this existence of the target interactors view. So now if you go to a disease associations page, you have this option to see interactors for a given target and all of their association evidence data for that specific disease. And I'll show you what that looks like later on. We've also kind of revamped our data downloads page. So if you go to it, you'll see that it looks quite different. And importantly, it's now powered by croissant, which allows us to kind of be fairer and more ML ready. We have revamped our molecular structure viewer on our target profile pages, and we've also added a molecular structure viewer to our variant pages. And what you see here is you can see the structure and the predicted structure from AlphaFold, but you can also see the alpha missense predicted score. And again, I'll show you what that looks like in just a second. We've added new data into our gene burden data source from the Broad CVDI Human Disease portal. And we've added new annotations to our pharmacogenetics widgets that give you a sense of the directionality. So that's all the exciting stuff that's in this particular release. But I also wanted to mention, since this is coming up quite soon, that on July 9th, we'll be deprecating OpenTarget genetics. If you weren't aware of OpenTarget genetics, you have nothing to worry about. If you used OpenTarget genetics, you'll find all the new data in the platform. So I mentioned that in March, we merged all of that data into the platform. If you do need access to the historic data, we will keep an archive on the EMBL EBI FTP, and within our documentation, there is information on how to access this. If you are currently panicking, please don't panic. There is help. So if you go into the platform documentation, we have a whole set of FAQs where you can see some of the questions that have already been asked. And I've also pulled together a lot of the questions that have already been asked on the OpenTargets community. So if you don't find what you're looking for, please feel free to post a new thread and ask your question, and hopefully we'll be able to help you sort out what you need. So I think that's everything I want to say as an introduction. I'm going to move over to show you the platform and what that looks like. And I'm just going to do this. Cool. Does that work if I do that? Okay. Give me 1 second. Okay. All of you, please feel free to interrupt at any point if I say anything that is particularly shocking or anything that you would like to add to the walkthrough. So to start off with, I want to show you what some of the new data from the GWAS Catalog update looks like. So I'm going to find a specific study, and I just need to copy it over. And actually, before I do that, one quick thing that I'd like to show you, we've added these filters at the top here. So in our search bar, if you are looking for something and you want to make sure that you find the right thing, you could, for example, say that you're definitely looking for a study, and that will filter down your results. So I know what I'm looking for, so I'm going to paste it in, and that is an osteoarthritis study, which was published this year. And this is quite an exciting one because it brings together 87 different data sets. So it's a meta-analysis. Sorry, I'm trying to find my notes. So it's a multi-ancestry GWAS meta-analysis, and you can see on the right all of the different cohorts that you have in this particular study. And that brings in quite a lot of different new data. So if I scroll down, you'll see that for this particular study, for this particular publication, we have 203 different credible sets over here. And you can explore these in more detail. I'm not going to do that just now, but you can have a look at all the different credible sets that we have for this particular study in more detail. And I mentioned in the introduction that the other kind of major study that was included in this update was a study published last year from the Million Veteran Program. And I think this is the largest multi-population biobank to date, which is hugely exciting. It's a study that's still ongoing, so the publication that we have is their first one. And in total, we have over 170,000 different credible sets from this publication alone. That includes the first evidence, the first genetic evidence in the platform for ESR1 and osteoporosis. So I'm going to look up osteoporosis to show you what that looks like. So on the I really need to learn how to pronounce these words. On the osteoporosis page, you can see that ESR1 is now the third top-ranked target. And I think below, in the previous release, it was much lower. And you can see that here we have evidence for GWAS associations, and all of these credible sets come from that study that I mentioned, the Million Veteran Program. This is quite exciting because ESR1 already had approved drugs. So if I go into the Kemble widget, you can see that ESR1 already had some approved drugs for osteoporosis, so estradiol, for example, but we didn't actually have genetic evidence in the platform that supported that association. So this study brings in new evidence. There is also other evidence. So, for example, Wnt1 is another example of GWAS association data for this particular association. So Wnt1 had other data, but it didn't have GWAS association data. So you can see that's all from the same study as well. The other hugely exciting update, which I said in the introduction, was this ability to see target interactors. So here, for example, you can see so let's say ESR1 as an example, you have this additional menu here. And one of the options that you now have is target interactors. So if I click on this, it opens this subview where you see the top ranked interactors by association score for ESR1, and you can pick your source of target interaction. So here, for example, the default is intact, but you could also choose reactome, senior, or string. The other thing to point out is we've set these kind of cutoff scores for the interaction scores. So here it's 0.42, but say, for example, you wanted a higher score, you could increase that and it would narrow down the available interactors. So you can interact with this as you would the normal association page. So here I've clicked on the GWAS association widget for ESR1, but I could also click over to target prioritization factors, for example, and see all of the information for these new targets in the same view. And the reason that we thought this would be really cool was because your specific target might have an association, but might not be the best target, or might not be, yes, you might want to see what other targets in the network are available. And you can now do that directly within this association view. So you can really quickly see whether within that network there's a target that might be interesting for your specific disease. Is there anything you wanted to add? No. Cool. In terms of kind of updates to the interface and to what you can see, you can now look at these new molecular structure viewers. So I'm going to switch over to, from here, I'm going to go to a profile page. So I've picked PLS3. So I'm going to go to this profile page, and you have this molecular structure viewer over here. So you can interact with this. The default, well, the first one that you see will be the AlphaFold predicted one, but we also have other options. So say, for example, you clicked on X-ray, you could see the structure that was determined via X-ray and interact with it then. So this molecular structure viewer already existed on the target pages, but we've now added it to variant pages as well. So I'm going to find you a variant, and I had found this one earlier. So you have the same thing. You can click on molecular structure, and you can interact with the structure here. So you can kind of drag it, move it around, zoom in and out to see what you'd like to see. And this is showing you where your variant is. So the variant that's of the profile page that you're on within this structure. What's quite cool about this as well is you can switch over to this different view, which is pathogenicity. And here what you can see is, so for the backbone, for everything except the variant, you see the mean alpha missense pathogenicity score for all possible substitutions at that location. So say, for example, if I hover here, for this particular location, it's telling me that there's a mean pathogenicity of 0.225, and so it's likely benign, and so it's in green. But if I hover over my variants, I can see that for this particular variant, the pathogenicity is 0.915. So even though the mean at that point is 0.4, it's telling me that this particular variant is likely pathogenic, and that's why it's in red. We have added additional data for genetic associations, and that in this case it's in our gene burden. So the example that I gave, which was one of the ones that they gave in the study, was YLP. So this is new data from the Broad CVDI Human Disease portal. This is a new study that came out recently. It features pan-ancestry sequencing from 3 different biobanks. So that's the UK Biobank, All of Us, and the Mass General Brigham Biobank. And in addition to the independent gene burden analyses that they did on each cohort, they also did a meta-analysis of rare variation across ancestries. And what we've done is we've mapped the study's disease terms to our ontology, and we've processed all of those results. And that's contributed new evidence for 520 different disease target pairs in the platform. One example for that was YLPM1. I think it was personality disorder. Yeah, so this particular association is a new one in the platform that was brought in by the gene burden data we've added in this release. And you can explore that in more detail. We've added links to the publication, for example, so that you can go and see what was done there. A couple more things. We've added pharmacogenetics annotations. So this is new annotations in our pharmacogenetics widgets from PharmGKB through work done by the European Variation Archive. So let's pick a target so that I can show you that. So if I go into the profile page for VKORC1 and I go into pharmacogenetics, so you have this new directionality column, which is here. And if you click on the entries in this, you have this drawer that opens up with different descriptions of the effect of the variant. So what you can do here is you can quickly see whether a particular genotype for the variant is associated with a requirement for increase or decrease of the drug dosage. And so that's most of the examples here, I think, relate to that. But it will also say whether it confers an increase or decrease in enzyme activity, an effect on drug toxicity compared to other genotypes. And we've included all the links to the publication so that you can see where we get this information from. Very last thing. We have revamped the data downloads page. So all of this information that you see here, you can also find in our data downloads. So if you want to access all the data directly, you can do so here. We have added these filters. So you now have these, each of the data sets has a category attached to it, which you can filter down. So I can click here and get only the ones that are related to target disease. And we have described all of our data following the croissant metadata standard format. So this will simplify how our data is used by ML models. It'll hopefully make our data more discoverable and more fair, which is something that we always try to work towards. That is everything that I wanted to include in the walkthrough. Is there anything that anyone would like to add about anything that I've said? Cool. In that case, yeah, absolutely, go for it.
So you can on these what are these
like the
widgets? Yeah, you can click on the schema.
Mm-hmm.
So from now on, each data set has not just the schema, but also a column wise description of what you can find in the schema. These small icons indicates if which columns in that data set consider the primary key, and also this symbol indicates it's a foreign key in another data set.
Mm-hmm.
Also showing which data set and which column that is a foreign key too. So it helps users to put these data sets into a better context for a more efficient use.
Mm-hmm.
So that's the only thing.
Yeah, that makes sense. Yeah. I'm going to stop sharing my screen so that I can see whether there are questions.
Nothing so far.
Okay. We'll wait a little bit because I think there's a slight delay.
There is a lag. Yeah. Please feel free to ask questions.
Yeah. What is the thing you're most excited about in this release?
The viewers.
Yeah.
It's an MVP, so we need to expand on more annotation, but I think it's gonna open doors for therapeutic hypothesis building a little bit in a slightly different way.
Yeah.
First question.
Yes.
Oh.
So thank you very much for asking a question. This is very exciting. So the question is, how is the interactor score calculated? Is it drawn directly from the external sources that you mentioned? Would you like to take this question?
Yes, it's coming from the sources when it's possible.
Mm-hmm.
Also, the predefined thresholds were also some sort of suggestions that these sources provide. But of course, the users are free to choose more stringent or more inclusive thresholds.
Do you want to? Oh, sorry. Yeah.
Out of the 4 sources, only 2 have scores. So Intact has a score that basically is not about the confidence of the interaction. It's more about the accumulation of evidence for that particular interaction. So I remember that's something that we discussed with the Intact team, and they were very, very clear about the fact that you shouldn't take it as a proxy for confidence. So generally, for example, the threshold that they suggested, 0.42, which seems very arbitrary, but generally it ensures that you at least have 2 high throughput evidence, 2 independent high throughput evidence, or at least 1 low throughput evidence, in a way that you ensure that you, in principle, should be reducing false positives as much as possible. Both Signor and Reacton don't have a score in the source, and String does have a score, which the best way to learn about it is to go to the String database because it's quite a complicated algorithm.
Do you want to talk a little bit about the different kinds of interactions, or?
Intact basically tries to capture physical interactions. So is the traditional way of thinking about interactions is basically you have 2 proteins that interact to each other. Reactom defines an interaction as being in the same pathway. So it might be that the 2 proteins don't interact physically, but one is upstream and another one is downstream in the catalytic process. Signor tends to focus, they have a bit of everything, but they try to focus on directional interactions, things like kinase substrate interactions, for example, and they are very rich on directionality. So you know that A activates B, which is something that is not necessarily available in the other databases. And String is the more is functional interaction. So it's the most relaxed definition of interaction. Is when 2 things might be functionally related, but they are not necessarily interacting physically. So it might be that they are co-expressed, they are correlated, they are cohabitating, coexisting, doing functionally something similar, but they don't necessarily need to touch to each other, let's say.
We
Thanks.
also expand evidence by homology. If you learn the interaction from other species, given a homology, they will propagate to other species as well.
Literature mining is included in the string as well. So there are multiple.
It's very, very inclusive.
Nice. There's a couple of questions. So one is, what if external sources are compromised? And there's a follow-up, which I think is related, which is, since all research papers are biased or compromised by AI, just to put those issues forward.
Oh,
That's a very thorny question, I guess. I guess, so yeah, go for it.
if they're compromised, we don't really do post-processing. If they're compromised,
It
Our
probably
data
depends on the source, right?
will be compromised. Yeah.
So you can provide the example of GWAS Catalog is a good example where there, I think we should be quite robust, right? Because at the end,
Well, but if they're compromised, it depends on the which compromise they're talking about.
I remember when I worked there and I was asking this, what happens if a PubMed got retracted with GWAS? There's no way for the curators to know that, oh, there's somewhere a paper in the database which got retracted. Unless it's someone raised attention that that particular paper is retracted, it stays. And if it stays, we, the consumer of other data sources, we don't really have enough knowledge to know. What we can do as a resource is, if a resource that we import, its entirety become questionable, we can drop it. But within a resource, it's very difficult to distinguish.
Process.
And also you can consider, even if you would have a list of retracted PMIDs, for example, unfortunately, there are quite many evidence where the same PubMed ID shows up. So it would be quite complicated to drop everywhere or from everywhere. I think that would be very, very useful, but it's not implemented.
every data modality is different, right? Like it's very difficult to manufacture a clinical trial, right? This clinical trial is an extremely regulated process. That we have clinicaltrial.gov, which is an official deposition system. It's extremely difficult to fake that. Genome-wide studies are relatively hard to fake in a way. It's still possible.
Still possible, but yeah, I think that's more robust.
I quite frequently read these are mostly comments on certain publications that they were not necessarily methodologically stringent.
Yes.
And that raises questions.
So that's why, for example, we rely more on, so we assign more confidence on things where we have access to the summary statistics, right? And we can reprocess and we can QC, and it will have more stars in our confidence system in the OpenTarget platform.
Mm-hmm.
A report in a particular in a particular publication, that's the thing that probably is the easiest to fake, right? When somebody says in a paper, we found this.
One study, one paper is more powerful.
So potentially our literature mining pipeline is more exposed to that kind of results. The good thing about the way we do things is that any user can decide, okay, then I don't want to trust literature mining. And you can move the slider where you downweight your literature mining, and then your associations will be based on your assumptions about what's high quality and what's low quality.
And this is one of the reasons why we've implemented that as well, right?
Yeah, before that existed, there was always this, oh, I don't trust this.
Yeah.
And I think over time that became a solution that I hopefully people is using. I don't know if they are using it, but I think it's very useful. But
yeah, please also feel free to add any feedback that you have about the release into the comments. That's also very, very helpful.
Maybe you can show how to filter in the.
I can, yeah, absolutely. Let's try this again. 1 second. Okay, so I need to get. Zoom.
So Shimon, what are you more excited about?
Do you want to also say what you're working on in the team? Because I realized we didn't introduce people in the team.
I'm from the genetics point of view, and I'm most excited about the new addition of the MVP studies. So
MVP being Million Veteran Program.
that's right.
You, Chintan?
I'm excited about the download space.
That's because you work on it.
Well done on your work. That was it looks awesome. It looks really, really cool. So yeah, well done. So what we were talking about, if you click on it, so if you're on a disease profile page, or on an association page, sorry, and you click on this column option here, this will open this view. And what you see here is the weight that we currently assign to each of these data sources within our overall scoring. And you can say, for example, you don't want anything that has gene burden data, you can move that down to 0, and that will automatically recompute all of the scores so that they now have a weight. So anything with gene burden now has a weight of 0. And you'll see that's changed the scoring here on the side. So that's one way that you could exclude one of the data sources, for example, from your results. I'm going to stop sharing for a second and see if there are more questions. Ah, yay! Sorry, someone just said thank you for the honest answers, and I'm excited about the OpenTarget project proposal. Thank you very much. We're very excited that you're excited. This is all very lovely. Cool. There aren't any more questions at the moment, so we can talk through some other things.
Javi can tell us what's most excited about.
Yeah, I mean, we've been working a lot. I'm the backend lead, and we've been working a lot on the pipeline and release of the data process. We're improving that part very much, and the time it takes to run the pipeline is much lower now, and it's becoming easier to do by people in the community. And hopefully, if you stay tuned, it will be even easier to run the platform anywhere if you want to tinker with the web and more stuff. And so we're trying to make it easy for the community to contribute to the project too.
This is probably a good point as well to plug the fact that in October we're running a hackathon to celebrate the 10 years of the OpenTargets platform. So we turned 10 this year, and there is a project proposal form currently open if you would like to suggest projects for the hackathon, either that you would like to do yourself or that you want other people to do. So you can, I'll try and put the link. I don't know if I can put the link in the comments, but it's another one of our events that we're running. So if you're on our LinkedIn page, you'll find it there. But yeah, you can have a look at that, and we'll be opening registrations at the end of the month. So if you'd like to come, we'd love to have you. We'll be running it in person, but also online. So yeah, please do join our hackathon. That'll be very exciting. I realize I've said exciting a lot. It's just how I vibe.
We also have a community page if you haven't
Yes.
seen it before, so you can, it's like OpenTarget's community.
Yeah.
Space, and you can write your feedback or ask your questions, and we're very interactive. So
Yes.
please be present there as well.
Yeah.
Can you tell us more about the hackathon?
What do you want to know?
People can propose projects now?
Yes. So the idea is the theme, we've tried to keep the theme fairly general. The theme is target discovery. Obviously, ideally, you would use something from the platform, so the data, the code, or something like that, and reuse that in some way, but we've kept it fairly general. So you're welcome to submit any kind of proposal that fits the theme. And
It's gonna be an hybrid
yes,
event.
yep.
And you're preparing surprises, right?
But we cannot disclose it. It's gonna be surprises, yeah.
But yes, I think the idea would, for us, it's a nice way to kind of see how people are using our data, the kinds of things they might be thinking about. And so one of the things in the project proposal is also whether you need anything from us. So what kinds of things you might need to be able to do your project. And if we can feasibly do that before the hackathon, we will try and help you out. Just make sure that those resources are available. There is a follow-up question about the weights. So I'm going to, so the question is, instead of drag up or down the UI component, can you access the weights directly?
You can with the
Yes.
If
So if you
click here, once you
cool, I
then
will.
you will see the API query
Mhm.
that you can run to get the data.
It's not working. Okay, so I'm just going to share my screen again to show you what we were just talking about. It's taking what? Nice. So my disease, my column options. Do you want to?
So if you click, well, let's say you don't trust GWAS associations, which probably not a good idea because there's the new one. But if you want to see the values for this, you can export the API query, this is another nice feature of the web, which is that you can check the query that every one of the widgets that we have in the web are making to the API, and you can make it yourself if you want to. And in here, you see the query, but you will also see down here the variables for the query. And in there, you will see that the weight for that one is 0, and you see all the data sources defined with their own respective weights. This you can see just as an example. You can see in all of the widgets. So if you go to any of these, you will be able to see the query that this table is doing to get that data. So that's useful too.
Yeah, absolutely. You can also see, so I think we document the weights in our documentation. So you should be able to see the weight that we give it and in one page if that's what you're looking for.
It's generally useful if you need to query a small set, but I think if you really need to do a systematic query across all the data, generally it's a lot easier to do it by downloading the data and manipulating it. Especially it will be a lot faster than just looping through the API and trying to get the data. That's why the downloads page, in a way, we identified there was that gap that a lot of people rely on the API queries to do programmatic access of the data when we knew it was not the most efficient way of doing it. So we are trying to increase the documentation of the data download so that more and more people download the data. And if you have any question, query of something you would like to do, you don't know how to do it, post it in community, or even now, if somebody wants to raise something, it will be, you can post it here and we can walk you through what would be the best way to do one individual query across the whole data set, or counting something.
So absolutely, and there is a follow-up question which is about, is there a one-page documentation for data access? I'm going to show you very quickly what the documentation looks like. So I'm going to switch to the documentation. So this is the OpenTarget documentation. You have here on the side all of the different pages that you have just to kind of try and orient you. In your case, if you're looking for data access, we have a whole section which is about data access and details the different ways in which you can access the data. So for example, here is our page on the dataset downloads, which kind of describes how you can do that, and it gives you some ways to get started to do that. There's also so all the data access kind of programmatically is here. You also have a description of the web interface and the different ways, the different pages that are a bit more tricky in the web interface here. But if you find something that's missing, again, please do tell us. We're very happy to update the documentation to add anything that you think we should add to it. I'm going to go back to the downloads. Nice. Oh, thanks for adding the community space. Yeah. So Annalisa's just put a link in the comments to the community space that we have where you can ask us questions.
Annalisa, do you want to comment the history behind some of our features? Like, for example, how they go through the feedback from our partners, things like interactors.
Yes. Yeah, that's a good point. So what we do, we are a consortium, right? Pharma and academia. And we really want to make of course, our community happy. But before that, there is a work between us and our partners, both pharma and academia, to make our features and to develop our feature in a way that it's useful and covers all the use cases we have. So what we've done, for example, with the target interactors, we've developed the feature with partners. So we have met them, we have gone through the use cases, and this was true also with the merge. So the merge platform we developed, we talked a lot with the partners, and we actually interviewed them to see which kind of feature or data set they would prioritize for integration. And what happens is that through loops of feedback, of course, we make our features better and better, and then we release them to the public. Now, I talk about our partners because we are a consortium, but of course, this also involves you, right? So we really want the community also to help us develop our features. So you can always, through the community or by email, you can always give us feedback, and we will incorporate your feedback into or your use case in a way, and we will incorporate this into our features. But just, long story short, we really, everything we release, you can be assured that it's been going through loops of feedback, and we really, really appreciate the community efforts, pharma community and academia. Was it what you wanted to?
I think it might not be transparent to someone, right?
No, that's
even
it.
regular users, they might not
It's
know
true.
where the things are coming from.
So this is the value of the partnership, right? We use feedback and we use experience of our pharma partners to make our tools as useful as possible.
There's another question. To what extent can we leverage external datasets for the OpenTarget's project proposal? I'm going to assume that you mean the hackathon, in which case you're very welcome to use external datasets as long as you are able to access them. So one of the conditions that we've set is that that your code and anything that you have, anything that you use has to be publicly available for the hackathon. So as long as the dataset that you use is publicly available, then I think that shouldn't be a problem.
So a massive change that we did was back in March, right? When we released the platform merged with OpenTarget Genetics. I think it's hard to project maybe to the users to understand the magnitude of that change. That was a massive effort over more than 2 years by the whole team to try to make that happen. But part of what we wanted to, part of the reason why we did that was because we thought that by doing this, we will be able to keep the GWAS information up to date. And the complexity of that pipeline is massive because the amount of compute and data that needs to go through to crunch it to actual gene target disease evidence in a way is massive. So I think one of the bits that is potentially more interesting is the fact that even in the 3 months that went from March to now, we got an increase, at least in number of credible sets of 1/3. So it's maybe that doesn't happen every 3 months. It's unlikely to expect that will happen every 3 months. But it's quite significant. And the intention is to keep doing that, to keep getting all the latest GWASs as much as we can. And that's also due to the amount of work that happens in the GWAS catalog, EQTL catalog, a lot of our data providers.
Yeah, they're doing great.
Considering how much effort went into just fine mapping to get these credible sets. Considering the old infrastructure, the old genetics portal, it would take years to get this release out.
I remember when we included FinGen in the OpenTarget Genetics, that was a multi-month effort to just get it right in the right shape, try to process it, colocalize it, well, file mapping first. Well, we didn't file map at that time because it was the file mapping from FinGen, but co-localizing N2G scores, all that's massive. So more studies next time.
Definitely more studies. We are expecting to gain some summary statistics from MVP that we were not able to harmonize this time. So
UK Biobank as well.
yeah, that's right. That's
Yeah,
right.
there is a discussion at the moment with the GWAS Catalog. They're actually integrating and harmonizing them. So that's gonna be a big addition as well.
Mhm.
Finance is true.
Yes, and we are trying to also include the larger meta-analysis between the MVP and UK Biobank as well. That's right. Keep in touch with this.
In case of these big meta-analysis, if we have both data sets that were got meta-analyzed, we have all three.
Yes, we can have all three.
We have already a significant amount of
Meta-analysis.
duplication in a way, right? Like of things that, so the same individuals being part of multiple cohorts.
Yes, the studies shows the cohort, and then you can see the same cohort popping up in multiple studies.
Especially UK Biobank.
Yeah.
But
This is why the GWAS Catalog wanted to include cohorts in there, because we like they see that the same people were analyzed multiple times, so that's transparent now.
Mhm. So how, I genuinely don't know the answer. So what the value there comes from the fact that you have multiple different analyses. So the fact that there are multiple analyses gives you a better indication that it's even if they are the same people.
They are analyzed most likely in different manners, right? So
Yeah.
Effectively, each time you add additional meta-analysis, you just increase the power, the statistical power of the associations that you get
Mhm.
as a result. So
Yeah.
you can be more sure that they are correct.
generally everyone is after power, right?
Mhm.
There's a bit a flavor on ancestries as well, like diversifying ancestries will potentially lead you to different signals. But yeah, power is the key. So most likely when you have new studies, they are the same as before, but something else, or you start combining biobanks. I think the osteoarthritis paper that you shared is a good example where basically you go to everyone who has catalog osteoarthritis, and then you put all these genotypes together and try to reanalyze it again. But yeah, there is a question for us also in terms of replicability and also like whether we are constantly reporting the same signals or not. But yeah, but you can say on the examples that you just show on the estrogen receptor, how we still find new signals that we didn't have from before that have a strong biological explanation.
In some sense, this also leads back to this previous question about the pause
Yes.
data.
Yeah.
That given we are integrating data from so many resources, even if there's some noise, you assume that all the combined directions are pointing to the right targets.
Yes, our study designs what matter the most, even though they are quote unquote essentially from the quote unquote same cohort. So I guess it's a question.
Study design as per GWAS.
I think it's a question about what we yeah.
It's rare to find the same, I mean, two publications doing the same GWAS or the same cohort twice, right?
So that's the thing that sometimes you have a publication, obviously, based on one particular set of people, and they are measuring. I know this osteoarthritis group, for example, and then they might run different analyses to stratify patients differently, select patients differently, because sometimes it's not clear what's the biological unit of the disease. So you can classify by how the osteoarthritis is developing on the cartilage, and you can classify them. And then it has an impact on the power, because if you do a clever selection, you get the higher power. And then in one publication, you can see the same people analyzed in many different ways. And then some yield significant results, other not. So it does have an
measurement.
Does have an impact.
So what we have in this particular example is different anatomical regions. Like you have the global osteoarthritis, and then there are studies of the knee, on the hip, I think.
But even just the knee, so just how the meniscus is, they are looking at X-rays, and people manually classifying X-rays, and they do the analysis based on this.
So yeah, study design is important.
Nice.
Yeah.
So Javi, technically, what's the next challenge?
He just did one release. Give him a break.
I think we're going to go.
Some people might be interested in.
No, it's a good point. Yeah.
Pipelines, things like that.
After finishing this upgrade of the pipeline that we are doing, which we are almost done now, finishing the part that generates the outputs. I think the next step will be to streamline the deployment, the architecture online, to make it leaner for the development part and easier for people who want to deploy it in the community, so they can run it anywhere or as easy as possible, and also make hours on anyone who want to host something, if they want to host a copy for any reasons, easier, more reliable, and also more easier to observe, like having better observability metrics and logging and information. And another step on that is to improve the process for the release, so it's less risky. Because currently we rely a lot on deploying the infrastructure on a new release, let's say, at the same time that the actual release is being done. And if anything goes wrong, then it's happening in the moment we're doing the release.
Nothing ever goes wrong.
Yeah. So it's not that much of just hitting a key and praying, but having something that is already there, and then we can switch to that once the time comes. So we're having more ease for everyone to run the platform. And
I think we're interested in knowing. I mean, we know there are many instances of the OpenTarget platform that people have internally, but for us, it's always useful to know if anybody is doing that.
Or trying to run Gentropy or any of the tools. Yeah, that's a good point.
That's another idea that I have, that is enabling some kind of call home, always opt-in, that people who set up a new platform or experiment with it can tell us there's one new platform in here, even if it's temporary.
Yeah.
But we use
It's optional, right? Some people will want to keep that private, but
It would be nice to.
when you're doing the setup, maybe the script that sets it up asks you if you want to report back.
Yeah.
we can always help some of that people.
Mhm.
Not all, but maybe some.
But yeah, it's always very, very cool when we see that people have taken the stuff that we've done and run with it and done other things. So we had the molecular interactions platform. Is it molecular? No, sorry.
No,
What am I can't-
childhood.
Yes, there was a specific yes, thank you. There was a specific platform, and I can't remember what it stands for now, but that took the kind of structure and added in specific data for cancer childhood. And that was, we had a great time seeing how they adapted it and that kind of thing. So please do tell us. We love hearing about these kinds of things.
And also it's easier for us to improve it for the people who use it, to make it easier. If we know what do they stumble upon and what problems do they have.
Cool. There aren't any further questions.
I'm very interested to see if anybody's using AIs to query our data.
Yes. I mean, there's some new stuff, right? Like a couple of people have posted in the community about some of the work that they're building.
Some of the MCPs.
Yeah.
Yeah, if people, because I guess a lot of people these days try to use AIs for everything. So anyone who has any, or even has done something or has anything in mind that they would like to do. I think knowing in community or reaching out, I think that would be quite interesting. Of course, we have our own plans, but yeah, we'll discuss that.
They can be adapted. Yeah.
First.
Yeah. Nice. I think we'll probably end there. Thank you so much for joining online. Thank you for participating in our experiment. I had a lot of fun. So thank you very much. Thank you very much for all your questions. Thank you all of you for joining. And yeah, maybe see you at the next release.